primers to find new enzymes within there. And I want to finish with two other case studies, which I think are probably the most promising things that we're currently working on. This is a strain of Nanoporopsis gatatana. This thing is an absolute beast. It grows extremely fast. And we sequenced the genome on it about a year ago. And it turns out serendipitously that when we look at this old model system versus this potential commercial system, that the genome on here is about a quarter of the size. It's much simpler to interrogate and to go through. And it is basically has the capability to undergo homologous recombination. So now we do have the tools for the first time to genetically manipulate this system. And this becomes a huge oil droplet. And very, very importantly, when we take nitrogen away from this organism, it doesn't quit doing photosynthesis. Now, red autofluorescence is the photosynthetic capacity, but it just keeps chugging. This great big huge oil droplet begins to grow out the back end, and ultimately we get this cell full of oil. It's got a very simple lipid profile, which is very good for converting into biodiesels. We can get half the biomass natively into the lipid fraction. And then we can go through this system. We just got the transformation established for it, demonstrated that we could do homologous recombination in there, have all the genomes sequenced in there. And now we can look at what makes this organism special relative to all the other algae that are out there using a series of Venn diagrams. And then we can begin to construct the different metabolic pathway maps. And the slide I wanted to cut down here to is this organism uses only chlorophyll A, and it uses carotenoids. It doesn't use any of these other pigments. And so it natively has this very truncated antenna, which is one of the reasons biofuels are so inefficient. Organisms evolved to be greedy, that if they encountered a photon, they were going to grab it and they were going to do chemistry with it. It's too big. If we cultivate them, we need to let them know that they can have a smaller antenna system for that light harvesting. This organism has a built-in smaller antenna. So the way the reaction centers are set up, we want this to be as small as possible, how many different pigment molecules are associated with it. And it's a very complicated place when we start to assemble these different pigment molecules and proteins. And most people go by knocking these proteins out to diminish those. But if you don't do that carefully, you get a very sensitive strain which becomes photoinhibited and starts to lose productivity. Take home on it is it's basically got an optimized photosynthetic light harvesting complex already. And then we can come in here, we can build the pathway maps to make our triacylglyceride droplet, our tag oil on the bottom. We can look at things like the carbon concentrating mechanism, how to get more CO2 to our Rubisco enzyme, and how to beat that up. We can come into the Calvin cycle and look at the breakpoints that we have in the Calvin cycle and think of ways we want to beef this up. And that's how CO2 gets fixed in the real world. And then ultimately in here we have a very good catalyst for light harvesting biofuel production. It goes into this intermediate stage, and then ultimately we can fill it full of this lipid droplet. And not only was it a good wild type organism, it turns out that it has got the best genome and the most sophisticated genetic tools we've worked with. So I think it's a very promising system. So I'm going to end with the next phase. Okay, C16, C18, not so great for biofuels. C14 is very good. C12, highly desirable. How do you get C12? So it turns out that 20 or 30 years ago, a group of scientists in California found some thioesterase enzymes. And these thioesterase enzymes are basically going to measure the length of your fatty acid. And when your fatty acid gets a certain length, it'll cleave it from the biosynthetic enzymes. And so if we use this C12 thioesterase, it'll cut off a fatty acid chain when it's 12 carbons long rather than the typical 14, 16s. So all we have to do is put a single gene into this organism and we change the lipid profile. 20% becomes C12. We want C12 because it's a better fuel. And so what this organism does, we put that gene in, it makes the C12, and basically 90% of it goes into the triacylglyceride. So now the fun starts. We said, okay, this is a better fatty acid. We put it into a diatom. A diatom has an oil droplet. It makes C12 and stores it in the oil droplet. Let's put it into a cyanobacterium. A cyanobacterium doesn't have a triacylglyceride oil droplet. It doesn't make triacylglycerides. What's it going to do? It's not going to want to put that fatty acid in the membrane, because if it puts it in the membrane will get too short. There's a reason biology uses C16 and C18. So we put the gene into a cyanobacterium, Synococcus 7002. We show that the C12 is produced when we put that single gene in. But then what we see is that the mutant secretes all of it. It can't store it, it can't put it in its membrane, so it just pumps it out. Now this is getting us closer and closer to the leaf now. And so, basically it's a detergent, right? So when you grow these in the lab, that's the wild type, and then these are the two expressing strains, and it starts to foam up. But now we got a new problem. It's great, we're making these short-chain fatty acids better fuel, the cell's spitting it out. We don't have to harvest cells anymore, but every contaminating organism on the planet gets into this thing and eats those C12s as fast as you make them. Reduced hydrocarbon plus oxygen is biofuel for the other organisms too. So now we need to be able to harvest that fuel before these other organisms get it. And we're working on basically that same magnetic strategy with these mesoporous resins, which will bind the fatty acids to amines internally, and then trying to pull those out. But it's a huge problem, and any insights and investigation and cleverness that people have, if you can somehow pull this out of the medium, there's going to be a huge, huge advantage and a big asset. The other place we're heavily invested is looking into these terpenoids. And these are going to be a much, much higher value product for us in the short term. There's already pre-existing industries for these products. The one that we're focused on right now is beta-caryophyllene, but we work on a suite of others. And so beta-caryophyllene is a very interesting molecule. When Calvin did a sabbatical in Brazil, he noticed that the natives would drill a hole in a tree and sap would run out. And then twice a year they would harvest about 20 liters of this sap, and it would run their farm equipment straight with no processing, nothing at all. He said, that's pretty cool. So he grabbed some of this oleo resin, he took it to Exxon, and Exxon came back and told him the majority of what he had in there was this beta-caryophyllene. And this has got all kinds of other uses. It binds to the cannabinoid receptor. It's responsible for some pain relief without the high, and so there's some interest in that. It's got some stuff in the heart disease literature as well. It's used as an aromatic. It's used as a spicy, sort of aromatic woody smell to different things. And so it's got a much higher value purpose other than fuels, but it turns out it makes a great fuel as well. It's got a cyclobutane ring. But all these isoprenes and these terpenoid products, they all have these double bonds associated with them. And so you can make really clever and cool polymers. A lot of polymer chemistry is ethylene, styrene, and vinyl. But you start to get these other groups, and you can do some very cool chemistry with this. And so nature puts these together, puts them together in blocks of 5. C5 is isoprene where you start, and we've made cyanobacteria that will grow and just basically bubble out isoprene, and then you can take that and polymerize it if you want to make rubber if you want. But again, it's coming out of the cell. The beta-caryophyllene is secreted from the cell. We have limonene, so we have our lemon fresh cyanobacteria now. And so where all this is kind of going is that the next stage of this is that how do you think about harvesting? And it's hard with the fatty acids, but maybe tractable. But this company, Algenol, came up with this idea that they're going to have these cyanobacteria. And what these things were going to do is they were going to make ethanol. And they were going to put a big plastic cap over them. So the cyanobacteria made ethanol, and the ethanol comes out with the extra heat that comes into the system, but then it condenses on the roof of this, and then it runs down to these gutters, and all this fuel comes flowing out, so they don't have to do any harvesting. They use the excess energy from light to do the distillation, actually in situ, and you can do that same thing with these terpenoids and these isoprene products, if you get just the right product with the right boiling point in it. And so in the end, we continue to make advances. We recently found genes that would make fatty alcohols and alkanes. Basically, you can do a step where you do a reduction with NADPH. You get a fatty alcohol, very good fuels. If you take that and you deformulate it, you get an odd chain alkane. We can put all these in, and we can get straight hydrocarbons out of the algae now. And at the end of the day, we can come back to our basic metabolisms. It's all about shunting and partitioning where we're going to traffic in there. And at the ultimate end, we have to come back and say, how real is any of this? We can make all these products, it's wonderful. If we look at where we are basically now with algae, it's this case 1. It's a very big footprint. We're very good, I think, right now, almost at case 2 in the last few years. If we get everything totally as efficient as it can possibly be, that's the footprint. It looks benign if we throw it in Nevada, but it's basically Lake Michigan. It's a big-time project, but if you look at how much corn we currently grow, it's amazing what happens when we put humanity onto something. I mean, it's a huge footprint that's actively managed. If we were going to get all of our fuel from the corn kernel, that's the footprint which we would require. require, which simply is not ever going to happen at these levels. If we're going to use cellulosic biomass, that's the footprint that would be required. And that's still very unlikely to actually materialize. So at the end, I think it gets back to algae. We have the assets where we can get into salt water. We can make triacylglycerides. We can shove metabolism where we want. We can get proteins. We can get carbs. We can get lipids. But most importantly, or most recently, we can get secretion. We can get different lengths of fatty acids to come out. We can get volatile terpenoids to come out. We can really start to shift these. And if we're clever enough to figure out how to harvest them, and if we're clever enough to figure out the last two big blocks, which are photosynthetic efficiencies by attendant truncation and CO2 assimilation, I think we really can put together something very, very interesting. And I think when we start to combine some of these different philosophies, we can combine very clever engineering, molecular biology, and genetics. At the end of the day, the sun does have enough energy to make this happen. Biology has the efficiency to transform the sunlight into chemical products. If we get this into Gulf of Mexico, someplace offshore, maybe it's not as troublesome. But we can do it. We can make our transportation sector with this. If we combine that with just some very modest and benign conservation efforts, I think the whole thing becomes infinitely more tractable. I think we could use probably half the fuel we use now without suffering any real dramatic lifestyle consequences. I mean, I think we could just be more efficient. And then just think if we did that, and if we have that thing, we could really be talking about a tractable solution. And if the transportation sector then becomes more electric, what we really want these liquid fuels for are jets. We throw that in half yet again, and then we're just down to a quarter of the size. So the technology is potentially there. It's going to take a lot of advantages, a lot of insight. But we can get way outside in our thinking. And I love this idea of putting hydrogen in at night, dumping out hydrocarbon all day. And we're trying to make these into artificial leaves now. And I was talking to Steve, and he's trying to break biofilms. I'm trying to make biofilms. But I want to sequester